And Mara's going to kick us off. Thank you, Jeannie. I'll turn it over to Mara quickly for the first slides.
Yeah, so like Jeannie said, Amy and I are going to do a quick overview of Eurasian Watermilfoil Biocontrol. You can go to the next slide. All right, so the biocontrol that is known about is the milfoil weevil, with the scientific name there that I'm not even going to try to pronounce. These weevils are native to Wisconsin and much of North America. And they have been documented to be responsible for natural EWM declines. And these weevils were also first studied in the 1980s as a biological control agent. So they came in not long after EWM became present in Wisconsin. Go to the next slide. There we go. So normally, since these are a native weevil, they normally live on our native northern milfoil. But as EWM comes into a lake, they shift their preference to feed on EWM because EWM does have fewer natural defenses and is more nutritious for the weevils. And then moving on to a little bit of the life cycle. So these eggs are laid on the growing tips of the EWM, so the apical meristem. So the larva will hatch, they mine the stem, so they're actually inside the stem of the EWM. And because they're mining in there, this does the most damage to the plant because it allows for pathogens and bacteria and stuff to get in there. And then the pupa develop within a actual pupal chamber inside the stem of EWM. And then moving on to adults, the adults feed on the leaves and lay eggs. And they kind of have, so in fall, the adults will fly to shore. So that's around September to October. And then obviously during winter, they're going to be hibernating. They'll be on shore in the soil or duff layer. And then spring, they're waking back up between April through May, and they're flying back to the lake to start their summer plans.
Okay, so next, let's talk about how weevils can help control EWM. What do they actually do? They cause damaged, blackened, or broken meristems, which reduces the number of flowers and seeds produced. And of course, remember that healthy stems are white or pale pink. They damage the vascular tissue with that larval tunneling. You can see how the larva has completely hollowed out the inside of the stem, including that pink vein that runs in the middle, transporting nutrients. Next slide. And then the pupal chambers, we can see this stem is positively peppered with pupal chambers. Next slide. We can see a whole bunch of them there. And you can see in the close-up pictures, we have an occupied pupal chamber that is completely occupying the center of the stem, destroying the vascular tissue. We can also see empty pupal chambers where you can clearly see that it is completely hollowed out the middle of the stem. Next slide. So the damaged vascular tissue causes a loss of buoyancy. Stems arch downwards. Stems can't reach the surface anymore. And that's how you end up with fewer ugly weed mats. This also causes a loss of nutrients. It opens up the plant to infection, and you have reduced health and vigor of the plants. So where do weevils struggle? This would be in sparse EWM beds, far from shore, in deep water. They also struggle in high disturbance, like from boats and harvesting. They struggle where there's lots of disturbed shoreline, like mowed lawns, beaches, paved areas, impervious surfaces. And if there's a stunted panfish population, this can cause heavy weevil predation. Where do weevils do well? They do well in dense EWM beds, in shallow water close to shore. They do well where there's lower disturbance from boats and harvesting. They love lots of natural shoreline, and they need a balanced fish community to do well. How many weevils do you need? This varies from lake to lake. There is no easy answer. It depends on conditions. The target level is 1 weevil per stem that we're usually looking for if we're stocking or if we're taking a survey of weevils to determine the population in the lake. We're usually looking for 1.0 weevils per stem to see a EWM decline or crash. However, as low as 0.25 weevils per stem has been documented for a natural decline in some lakes where conditions are perfect. How is the data collected? There's a couple ways you can do it. The volunteer monitoring protocol is part of the CLMN AIS monitoring series, and that would be chapter 12 of the CLMN AIS program. There's also professional level monitoring that we as professionals can do. And you can find that in the Biocontrol manual. The QR code there can take you to both of those resources. Stem samples are collected in tandem with the aquatic plant survey. So when you're doing your PI survey, you can grab some stem samples. And then the lab work is done using magnification to count the eggs, larvae, pupa, adults. What does success look like? We all know that biological control is not the same as eradication. What you see is shorter plants with fewer ugly weed mats, right? Less vigorous plants, so the natives can compete again. The EWM beds get more sparse. So essentially, EWM is not gone, but it plays nice with its neighbors. It is interspersed and not dominant anymore. How to use biocontrol? We can break this down to 3 basic options. Option 1, do nothing. Option 2 is adjust your management plan to support existing weevil populations. And option 3 would be stocking, as an artificial stocking. So option 1, go ahead. Thank you. Sometimes conditions, the option 1, do nothing. Sometimes conditions are ideal, and simply waiting for nature to take its course really is the best plan. Sometimes resources are so limited, there really is no other option. Like if there really is no lake group there to take action. This option requires patience, so lake residents need to know enough information about biocontrol to feel confident waiting. Our example that we have today is Curtis Lake, and Mara's going to take the next slide.
So Curtis Lake, we did a site visit. So a little bit of background on the lake is it's a 42-acre private lake with 1 small inlet source, as you can see on the right, but it's a very small inlet source. It's about as much as a garden hose. And there was limited support from a lake group, which I'm not even sure if you can consider it a lake group because it was just one guy who really cared about the lake. And Shelby Adler was able to confirm EWM there, so we plan to visit the lake. So what we found when we visited is that we did find dense EWM throughout the lake, which we found was really great for nursery sites. And we were able to bring some samples back and were able to find the weevils in all life stages. So they had probably been there for a bit of time. We had found some evidence of weevils at work. So we saw a lot of damaged vascular tissues and kind of wilting EWM under the water. And I think the main takeaway from this visit was that the landowner is now knowledgeable about the biocontrol, and he's going to continue monitoring, and he's really excited about this. You're muted, Amy.
Thank you, Mara. Emily, can we go back one slide? I was going to make a mention of that photo. So Jack is the volunteer who's going to monitor, and that's his photo actually. One of the ways he's going to monitor is by taking a photo the same time every year, looking the same direction. So he's standing on his dock and he's going to take ones in each direction and label them so he knows the date and which direction he's facing. That's one way to monitor for weed mats. And in lakes where ugly weed mats have been a problem, you can take basic monitoring using photo documentation to compare from year to year. That's been valuable to me over the years, a really simple thing volunteers can do. Okay, next slide. So option 2 then, support weevils. How do we do this? Biocontrol can actually be integrated into many management plans. And a few ways you can do that is by improving shoreline habitat. Buoy off important weevil nursery areas. You can limit harvesting in important weevil nursery areas. You may also be able to reduce predation pressure. So looking at natural shoreline habitat is winter survival for them. And of course, this is one area where lake residents have control and can take immediate action. It's also not that hard and doesn't cost that much. And that 35-foot buffer that we are usually recommending for lake health, that's for weevils. So weevils are just one more reason that all plans should promote buffers and work on improving natural shoreline. Good habitat. My research found that it's really quite simple. Just 1 centimeter of duff material or more, that's all they're asking. And it doesn't matter if it's leaves, sticks, grasses, wildflowers, pine cones. They just need a little something to hide under for the winter. Bad habitat would be mowed and raked lawns, beaches, wet soils, but mowed areas can still be okay. Think of it as time sharing your shoreline with the weevils. If you mow only from Memorial Day to Labor Day, and then after Labor Day, you stop mowing and let the grass grow, let the leaf litter sit there in fall, and wait until Memorial Day to clean up the leaves, start mowing, that gives them that safe space on the shoreline to hibernate for the winter and then come out safely in spring and get back to the lake. So buoying off weevil nurseries, we would do this to prevent boat traffic from cutting off those all-important growing buds, the tops of the plants that are so critical. It's easy to do and it really doesn't cost that much. The weevil nurseries that you'd be buoying off would be those dense beds in shallow water, close to shore, where boats probably shouldn't be ripping through anyway. So it's a decent option. Go ahead. And limiting harvesting in weevil nurseries is very similar idea. We're preventing the harvester from cutting off those all important growing buds. It does take planning, but this shouldn't cost that much. And the weevil nurseries, again, that you're looking to avoid would be those dense EWM beds in shallow water, close to shore, where the harvester probably shouldn't be going anyway. What about reducing predation pressure? We actually did an experiment, a pilot study on Lake Jonas, using predation exclusion cages to adjust for a stunted panfish population. The pilot study at Lake Jonas had excellent results, very exciting. It does cost money to build the cages, but they're reusable from year to year. The labor was pretty minimal to place the cages in spring, leave them alone all summer, and then just remove them in fall. So it was elegantly simple to boost the weevils this way. But there are some caveats. First, we'll answer that question. A stunted panfish population would be where you have lots of those little panfish, sunfish, bluegills, and not a lot of big predator fish. So if you've ever gone fishing and you get frustrated because all you're catching are those little tiny things that are worthless as far as keepers go, and you can't find the big ones, you might have a stunted panfish population. That would be an example. So the caveats, it does require really specific conditions. EWM beds in shallow water, 3 foot or less, because of the height of the cages. If you want to build them bigger for deeper water, they're going to be even heavier. The firm sandy bottom was needed because we walked them in and walked them out. They're heavy, they're awkward. And this pilot study should be tested on more lakes to determine whether it can work on more lakes, or is Lake Jonas just an anomaly. But it was exciting results with an elegantly simple design, and I'd like to see it tested on more lakes. Next slide. Option 3 would be stocking. And this costs more. You have to contract the starter stock, so that there's a considerable price tag there. It requires a lot of volunteer labor because then you take that starter stock and you have to do the mass rearing program to get your numbers up, what you really need for stocking into the lake. So lake groups have to be committed to the cost and the labor, and commit to a 3-year trial, because if you're only going to try it for 1 year and then give up, you don't really give it time to see if it's working. And Kathy Higley will present on this next. So there's the screen with my contact information and Mara's. We have a QR code for the resources that I talked about, the monitoring protocols, Weevil ID information, and the Lake Jonas experiments. Our website, our Biocontrol page has a lot of that information for you.